Hello everyone, I am Shubroto Sharkar from Bangladesh. I will share the findings of a GlobalSeaweedSTAR funded research project in Bangladesh on spatial planning for seaweed farming in the maritime zone of Bangladesh. Bangladesh is a maritime country with a vast maritime area which is enriched with diverse species diversity. And among this species diversity, we have many species of commercially important. And among these commercially important species, seaweed is one of the promising component for blue economy in Bangladesh. At this moment, we have more than 200 seaweed species, and 10 of these species are commercially important. However, few farmers are involved with seaweed farming in Bangladesh. There are several reasons for that. For example, we do not have any habitat map for seaweed farming in Bangladesh, and then we do not have any information on seaweed disease. Farmers encounters a problem with the disease, so they face the problem with the crop loss. And since we have to export the seaweed to the foreign countries, but we do not have any information about the food security. Therefore, this project aimed to identify the suitable sites for seaweed farming in the maritime area of Bangladesh, and then verify site suitability by comparing the seaweed production data from existing seaweed culture farms, and then generate special information on seaweed pest and disease. Finally, monitor chemical composition and heavy metal concentration in Bangladesh, in seaweeds of Bangladesh, to ensure the food security. So this is the method for habitat modeling. So we used different types of data, like in situ data, satellite data, and model simulation outputs, and we considered several variables, and then we performed some statistical analysis like multi-collinearity test, and then we used generalized additive model to predict the habitat of seaweed, and then we used predicted probability of seaweed from the model, and then we compared this probability with the seaweed production data from different farms to validate the model. And then we considered 5 commercially important species for disease, metal, and bioactive compounds monitoring, and the methods are described here. So this is the findings. So this is the response of seaweed to environmental variables. This is the output of the GAM model, generalized additive model. So here we can see seaweeds can grow the moderate turbidity, and then shallow water, and then high nutrient concentration, salinity, they require high saline water, and current should be moderate. And this is the spatial prediction of seaweed habitat, and here we can see about 11,200 square kilometer area had the probability of 20 to 100% occurrence probability of seaweed, and 4,100 square kilometer area had the higher probability of seed occurrence, that is 50 to 100% occurrence probability. And most of the suitable habitats are located in the southeastern coastal zone, while moderate area is located in the southwestern coast, but central coast is not suitable for seaweed farming because of less saline water and high turbidity, and this is due to the high freshwater flow from these river systems. And so we compared the predicted probability of seaweed with the production data from seaweed farms, and we found that our model has a good agreement. So our model is a very good predictor of seaweed habitat in Bangladesh. And we analyzed the disease, and this is the first record of seaweed disease in Bangladesh, and we found 7 types of disease, and among these disease, epiphytes is the most common types of disease. And among the epiphytes, so we found small filamentous algae, larger macroalgae, and biofouling organisms are the most common epiphytes found in the seaweed of Bangladesh. And this is the spatial distribution of seaweed disease, and we found that southeastern coast has the higher prevalence of seaweed disease. And we analyzed 13 elements, metal elements in seaweeds, and we found that metal concentration are below the threshold level of human health risk. And we evaluated the mercury selenium ratio, and we found it was 0.10, and we also analyzed THQ value, and it was 0.25. So all this suggests that seaweeds are safe for human consumption in Bangladesh. And then we also analyzed the bioactive compounds and proximate composition of seaweed, and we found that seaweeds in Bangladesh are rich with energy, carotenoids, the DPPH, phenolics, and they have higher content of ash, protein, fat, and fiber. So these are the scientific outputs from the project, and based on these outputs we developed a user guide for seaweed farmers. And in this user manual we try to translate the findings of this project in a smooth way so that local farmers can understand. So in this user manual we provided information on seaweed farming methods, seaweed disease, how to monitor disease, how to monitor environmental condition, how to ensure the food security, and of course how to use the suitability map that we developed in this project. To disseminate our findings with the stakeholders, we organized a workshop. So in this workshop we invited seaweed farmers. So with this, when we invited farmers, we emphasized on women farmers also. And then we invited the government officials as well as the entrepreneurs. So we shared our findings with them, and then we asked them to provide their opinions, and based on their opinions we made some. So now we have a special habitat suitability map. We know seaweeds are safe for human consumption. We know what are the disease, but at this moment one thing is missing is like marketing channel for seed. So farmers, entrepreneurs, and government officials, they all said at this moment Bangladesh should develop a value chain for seed, and then we need to focus on product development. That means seaweed derived product. We should think about the innovative culture techniques to maximize the production. For seed problem, we need to develop gene bank, tissue culture lab, and then of course we need an information hub for seed farming. So all these outputs are published in scientific journals. We published 2 articles, and one article we described the seaweed habitat maps, and another article we discussed about the disease, metals, and bioactive compounds in seaweed. So thank you.